Good afternoon, ladies and gentlemen. Thank you very much for bearing with us. Today I have the pleasure to have with us David Fairen-Jimenez. And I would like to take this opportunity as well to thank you, David, for accepting our invitation and share a little bit of his research at the University of Cambridge. So David is a Royal Society University Research Fellow and a reader in molecular engineering in the Department of Chemical Engineering and Biotechnology at the University of Cambridge, where he leads the Adsorption and Advanced Material Laboratory. His research into the application of metal-organic frameworks, MOFs, in energy applications and nanoscale drug delivery is underpinned by fundamental studies into molecular recognition and adsorption processes in nanoporous materials. So it's wonderful to have David with us, who will share a little bit of the work that he's been conducting in Cambridge. And without any further ado, I'd like to hand it to you, David.
So thank you very much, Savio, for the kind introduction. So as you said, I want to give a couple of examples about the recent research we have done at the University of Cambridge. And in particular, I'm going to talk about this topic that you can see here in the slide, which is how can we really use the different tools that we have on computational and experimental techniques for materials discovery and molecular engineering of metal-organic frameworks. I'm going to focus about, as I said, computational research, experimental research, and I will focus quite a lot of ideas into the drug delivery by the end of the presentation. Okay, so just a general slide that I generally use to give a little bit of justification about the kind of work, the research we do at my lab at the University of Cambridge. I'm including here the projections for the population growth, and you can see here how many people are living on the Earth, okay, billions of people versus the years. And I have highlighted here two main dates. The first one is the end of the Second World War, okay. At this point, we started increasing from 2.5 millions of people into something that was much, much higher, okay, accelerating quickly. That was very probably because of the development of vaccines, antibiotics, different improvements in food industry, and so on. And this is where we are now. Okay, we have about 7 billion of people, and we have different projections included here. The most realistic one is this orange one that you can really see here, that suggests that we will end up this century with about 10 billion people living on the Earth. And therefore, we are going to focus, we are going to have very important challenges in this century. All these challenges are related with exploitation, of course, of natural resources, including sweet water, but also challenges in healthcare, okay, having so many people living on the Earth, an aging population, also with the food demand, obviously, and we are talking quite a lot about energy and pollution these days, especially with the COP conference in Glasgow that is taking place, as I said, these days. In my research lab, we try to cover most of these challenges that I have included here, the ones that I have highlighted here on the right of the slide. And this is also a nice slide to illustrate the importance of all these different challenges. And these are the century global challenges for this 21st century. And as I said, we work on applications that are related with healthcare, okay, in particular with drug delivery. We work on clean water and sanitation, water harvesting, pollution, hydrogen storage for affordable and clean energy, and also carbon capture for heating and air conditioning. Okay, a lot of different applications with materials that we are developing in our lab. And we work with these nanoporous materials, that we call from the 21st century. We have worked in the past with advanced activated carbons, also with zeolites. We have worked a little bit with mesoporous silicas, and we are definitely working quite a lot. We are dedicating quite a lot of efforts on these supramolecular materials, these porous coordination polymers that are called metal-organic frameworks. All in all, from all these classical porous materials into newer materials, the basic idea that we try to develop is to fully understand what is the physical chemistry which is involved in the absorption phenomena of different molecules, either gas molecules or drug molecules, so we can apply all this fundamental knowledge into specific industrial applications. Okay, industrial problems, this is basically what we are aiming for. And on this, we include some ideas on the modeling, okay, through Monte Carlo simulations, molecular dynamics. We also move into process modeling and machine learning, okay, a lot of different ideas about how can we really use computers to select optimal materials for a specific application. And once we are able to identify these materials, we really go to the lab, we work on material discovery, synthesis, we try to engineer these materials into something that we can apply on these different applications. Okay, if we are thinking about drug delivery, obviously we need to focus on something that must be biocompatible and also something that we can translate to clinic. And then we go into the engineering side of things. We obviously need to dedicate attention to scale up, okay? And if we are thinking about drug delivery applications, we need to think about good manufacturing practices, so something that we can really prepare on a sterile environment, but and also how can we really optimize the whole process of creating these materials and the efficiency or the performance of these materials in the different applications. I said we are working with metal organic frameworks. Okay, for those of you that doesn't know what is a MOF, basically a MOF is a porous coordination polymer which is created by the self-assembly of some metallic atoms or ions or clusters. Okay, here these are represented by these red spheres, and these are connected by these blue molecules that you can see here in the slide. And at the end of the day, this self-assembly will create these three-dimensional structures that you can see, and very importantly, you can see between all these molecular Lego that we are creating here, okay, between all these different building blocks, we are going to have a lot of space, which is something we will be able to exploit for gas absorption or drug delivery applications. Okay, this is where we can really host gas molecules, and we can control the absorption and the release of the molecules that we are including in the porosity. These materials are really important because these are crystalline structures that work like a molecular Lego, as I said. And basically, we are going to have a very precise control over these structures. Okay, just by deciding what building blocks we are going to use, we will be able to tell what is going to be the structure we will create. These materials also present extremely high pore volumes and surface areas. And when I say high pore volumes and surface areas, I'm talking about several thousands of square meters in a single gram of a material. So yes, a single gram will have a huge surface area that we can exploit for the absorption of molecules. We can also play with the pore size of the structures. Okay, it's very easy. Just having a longer organic molecule, we will be able to have larger porosity, and this is something we can exploit, obviously. And we can also do some post-synthetic modifications. We can include also into this external surface that we have in the material, for example, we can include different molecules. We can include polymers, we can include peptides. It's a lot of flexibility in terms of synthesis. And there are thousands of materials for any application. Just in the last 20 years have been published more than 100,000 different materials, as you will see in the next slides. The materials are not perfect, okay, like any other material. They will have their own challenges. Low mechanical and thermochemical stability is a big issue. And sometimes, for some applications, that's not going to be a problem, okay? In particular, if we are thinking about drug delivery, that could be something. It's not necessarily a challenge, okay? It's a challenge if the material dissolves completely in water, for example. But if the material is more or less stable, okay, until we release the drugs, but then we are able to dissolve the mob, that's going to be really good because we will be able to avoid any accumulation of these nanoparticles in the body. And then we will avoid toxicity, we will avoid side effects, and so on. Low density is a problem for energy applications, gas storage, but not really for drug delivery applications. And we have another problem, which is the fact that we have thousands of materials. So which one are we going to use? Okay, 100,000 different materials. How can we really find the optimal one? The benefit that we have is that all these materials are crystalline. Okay, we know the position of every single atom of the structure, and this is something really useful because we can really run Monte Carlo simulations, computational methods to really be able to screen all these porous materials. And this is a question that we are trying to solve. Can we find a deterministic predictions for materials performance? Okay, this is something that we have been working on. And also, can we translate these predictions to real materials and applications at scale? So what I'm going to do is I'm going to explain different topics, different ideas where we have been working, trying to see if we can really get an answer for these two questions. Okay, so this is basically what is included in the next slides. What you can see on the screen is basically how these materials look under the microscope. Okay, these are micron-size metal organic frameworks from a beautiful material called C8. And you can see all these crystallites, they have all of them, they have the same kind of structure, the same morphology with these square windows that you can see here, but also these hexagonal windows that you can really see, all these facets that you can see in these crystallites. Micron size, but we can really control the size of these materials from the nanometer to the micron or even millimeter size. For gas storage, basically on the left, we can accumulate one molecule. Will be the same if we are working about drug delivery. We can really accumulate and concentrate the molecules that we are trying to absorb inside the porosity. Whereas in the case of a mix of different gases, we can also talk about just one of the components being concentrated inside the porosity, but not the other one. There is an endless number of applications for these materials. Okay, all this is really related about the different challenges that we have found, the global challenges. We can really use these materials for gas separation, gas absorption, drug delivery, okay, but also for sensing, energy production, catalysis, multiple different possibilities with all these different materials. So, going to this question about what move for what application, right? That was one of the first questions that we were trying to answer, and thinking always on these deterministic predictions of this material. And for this, we were thinking about this question about what is a MOF and how many MOFs have been published. Okay, we can really understand this question, and if we have a database with all the materials that exist, the idea is that we will be able to use supercomputers to start screening all these different structures, right? We are not alone in these questions. There's always a lot of people working on these ideas before us. And we also need to be very thankful to the Cambridge Crystallographic Database Center, which is an institute that is sitting half an hour from our department these days, but it used to be just five minutes walking. And it's an institute that was created in the 70s, and they were trying to develop a curated database with all the crystalline materials that were available in the literature. So as I said, mid-70s, okay, they started, and they have accumulated a database that a couple of years ago, they were able to reach 1 million structures, crystalline materials, okay? Not only MOFs, but also zeolites, drug molecules, anything that is crystalline and is published in the literature goes to this database, and they are basically curating this. As I said, we were not the first ones trying to answer this question about how many MOFs have been published, and one of the first examples, okay, it's not the only one, but it's this work from Professor David Soll and Professor Randall Snarr. They published this in 2014, and they were looking at all these materials, and they found that in 2014, 5,000 materials were published, 5,000 metal organic frameworks. We were happy with this because obviously this is extremely useful, but we always thought the number was much, much larger than that. So we started asking these questions about how many MOFs have been synthesized, but also how many MOFs can be synthesized, okay, in the future. It also goes about this main question, which is, okay, can we really predict the performance of these materials? And also, is there an optimum structure for a given application already there? Okay, this is the key question that we are trying to answer. And when looking about all these different ideas, we went to the symbol that you can see here on my screen. This is basically using the tools from the Cambridge Crystallographic Database Center. Okay, this is something called Conquest, which is a tool that will allow us to explore the database with some of the ideas that I will start including in this slide. Okay, the explanation, the full explanation of what I'm going to have in this slide is in this paper that was published last year. Okay, it's a tutorial that is explaining exactly the process that we have. But basically, the idea is that we start selecting specific materials, and we were looking for different criteria that will allow us to filter what is a MOF and what is not a MOF. So what I have here on the right on the screen is a prototypical material called MOF5. It's one of the first materials that, one of the first MOFs that was published in the literature. Okay, it's from a paper in 1999. This is the metal cluster. It's made of zinc and oxygen. This is the organic molecule, an aromatic ring with two carboxylic groups connected with another zinc oxide metal cluster. And this gray sphere is basically representing the porosity of the material. Okay, so as I said, using Conquest, we take this idea and we draw this kind of schematics. Okay, basically what we have is the zinc atoms connected to a carboxylic group, the aromatic ring, another carboxylic group connected to the zinc oxygen metal cluster, and so on. Okay, all this is growing on polymeric situation. Basically, all this is translated using Conquest into this idea that I have included here. Okay, this is any metal connected with the different atoms, basically following this idea that I was highlighting coming from the MOF5. So this is just for one criterion. Okay, this is for carboxylate derived metal organic frameworks, but we were expanding this into different ideas. We were having different materials, maybe with formates, maybe with cyanides, maybe when we have connections not through carboxylate groups, but maybe connected through nitrogen or sulfur. All these different possibilities, okay, and this is quite important, all this is very flexible. Why did we use this criteria? Basically, because we were just comparing this with existing metal organic frameworks that we were familiar with and trying to see that we were able to cover all these different materials and not missing anyone, okay? This is flexible, as I said, and this is something that if at some point we have missed some materials, we can always adapt these criteria. We can modify them in order to be able to update this. Because at the end of the day, what we are developing is, with Conquest and the CCDC, the Cambridge Crystallographic Database Center, a curated database for metal organic frameworks. We were beyond that, beyond finding how many MOFs we had, and we also started paying attention to the different families that we have in this database. Okay, this is basically families made of zirconium or some materials that have a lot of open metal sites, and this could be interesting for some specific applications. Also, another kind of material called zeolitic imidazolate frameworks. We can also include zinc oxide-based materials or copper-based metal organic frameworks. Okay, different families, and again, all this is completely arbitrary. This is something that we can expand if we want. Okay, all the details as well. All this was published last year, and all this is really available in the literature. Very important. You remember the previous database I mentioned. Okay, published in 2014. They were talking about 5,000 different metal organic frameworks. When we published this 3 years later, we found that there were 70,000 different materials in the literature, and there were about 10-15% of them were porous. Okay, today we have more than 100,000 materials, and what is important, we have more than 1,000 new materials, new metal organic frameworks per month. Okay, so this number is really increasing really fast. Okay, now we have the database. So we have a list of materials, and we are always thinking about this prediction of the material's performance. Okay, and for this, we always rely on the first step on the use of molecular simulations. We are trying to answer these questions. What if scenarios? Okay, can we know if we take a material how much gas molecules or drug molecules will be absorbed inside the porosity? Okay, do they fit inside the porosity? We can use Monte Carlo techniques to answer this question, to find selective materials, but also molecular dynamics to answer this question about how fast drug molecules or gas molecules get absorbed inside the porosity. We can also get a lot of output information that is comparable with experimental data. And something very important, we can also get a very detailed picture of the absorption process on a molecular level. Okay, this is quite important, quite interesting, because then we can really understand how the materials behave. And then this is something that will allow us to improve the properties of the materials. This is one example of the simulation, because this is the question: do the simulations work or not? Okay, so before talking about drugs, I'm going to talk about gas absorption just a little bit. This is a material called NU1000. This is 100, sorry. This is coming from Northwestern University. They have these copper metal clusters that you can see here. And all this is connected with this organic ligand, a very, very long molecule with carboxylic groups. And these spheres that you can see here in the porosity are basically representing the different cavities, the 3 different cavities that you have in this material. So using molecular simulation, we can focus on hydrogen absorption, 77 Kelvin. It's interesting for energy applications. I have here the uptake of hydrogen, okay, milligrams of hydrogen per gram of sample when I increase the pressure. And you can see here the isotherms, the experimental one and the simulated ones. And you can see a really, really nice match of the experimental data when we were running these simulations. And it's important to highlight here that this is not a fitting of the experimental data. Okay, this is something that we have run independently, and then we compare face to face, and you can see that we have an excellent agreement between experiments and simulations. So really, really good news, because now we can really start working not with one single material, but with a full database of materials, right? We can really work with 100 different structures, and we can get these kind of maps that you can really see here. This is basically for hydrogen absorption on 16,000 different metal organic frameworks. And what you can see is grams of hydrogen per liter of material as a fraction of the porosity. And every single point you see here is the result of one single Monte Carlo simulation. Okay, this is something that we can run in about 1 month's time. Okay, but if you try to do this experimentally, obviously that's going to be impossible. Okay, this is synthesizing 16,000 different materials and running 16,000 different absorption isotherms for hydrogen. Anyway, can we find a deterministic prediction for materials performance? Okay, that was the main question. What I really want to have is not a Monte Carlo simulation. I don't want to spend 1 month running these simulations. What I really want to have is, if I understand what is the surface area, the pore volume, the pore size, density, and composition of the materials, can I really understand what is going to be the loading capacity of these materials? Okay, can I really predict this if I am able to map a landscape of properties and performance? So then we start moving into these drug delivery applications and healthcare applications, and still working on the gas domain. Okay, these are some results for oxygen storage. Okay, this is important for some applications we wanted to develop, and these kind of screenings, Monte Carlo screenings, allow us to understand what are the physical limits of all these materials for this application. What I have here is 3,000 materials represented. Okay, every single point is the result of one single Monte Carlo simulation for oxygen storage. And you can see how much gas we can store per unit of mass, okay, and how much we can store per unit of volume. This is at high pressure, okay, so is applying 140 bar pressure. But what we can really see is that optimal materials are going to be applied with a specific heat of absorption, which is basically related with the interaction between the oxygen and the metal organic framework. And this is good, but it's not too good, because at the same time, okay, if I say optimal heat of absorption, optimal interaction is these values, we have very good materials, but we also have really, really bad materials here. So why this big discrepancy? Okay, why this big difference? This is something that is clearly shown in this screen. The difference, the problem is that this is a multidimensional problem, okay? And if we only pay attention to one variable, this doesn't work. But as soon as we include some colors into these different representations, you can really see something that works much, much better. We need to have not only an optimal interaction with the material, we also need to have an optimal void fraction. And this is particularly important when we are talking about volumetric capacity of these materials. How much material, how much gas we can absorb per unit of volume. We run these Monte Carlo simulations, but we also want to have something which is highly dynamic. Okay, something that doesn't work for us and for anybody is to be able to run all these thousands of simulations, okay, and just to store this data in a research paper and just to have a figure where people can really see the values, because it is impossible, okay? I can highlight one of the best structures here maybe, but if you are interested in all this other data, that's going to be very, very difficult to really visualize and play with that. In order to do that, to facilitate this, we work with these interactive websites, and this is something you can really explore here. And basically, this is something that will allow you to change the different variables that you have in the different axes, y-axis, x-axis, the color, and the size of the symbols. The number that you see here in gray, this is the pressure, okay? But basically, we can really have these dynamic representations. And this is what is basically shown in this slide. You can see we are increasing the pressure, and you can see how the different data, the different individual metal organic frameworks are growing when we increase the pressure, okay? We can really see how the performance is changing when we are increasing the pressure. And this is important because this will allow us to really be able to understand different ideas that we have with these materials. For example, the fact that these materials, the purple one, they will continue growing when we increase the pressure, whereas these orange materials that we had here, they were stuck at much, much lower absorption capacities. This is how the Explorer looks in reality, okay? This is basically something that will allow you to play with a 2 or 3-dimensional representation of these materials. We can also play with the pressure, so we can really change the different properties of all these different metal organic frameworks, the properties at different pressures, okay? We can filter the materials, so we can really select different materials based on the names. These names are coming, by the way, from the Cambridge Crystallographic Database Center. We can filter them depending on the different properties or names. As I said, we can move to 2D or 3D representations. If you click display, you will see the effect of increasing the pressure, okay? What is the impact? And you can also change all the different variables, as I said, okay? The different axes, the color, and the size of the symbols. And what is important is that when you click for one of these structures, you can really see not only the properties that we were simulating, but also you can see here, this is the Cambridge Structural Database link. If you click here, you go to the original paper, and then you can really see the properties of these materials, the composition, and the synthesis as it was reported in the literature, okay? So very useful tool. And just to finalize these couple of ideas, yes, we can also confirm the fact that the simulations and experiments, they match each other, okay? The experiments are able to simulate the experimental data properly. This is for the nitrogen isotherm, just to check the quality of the material, and this is for the oxygen at room temperature. This is the comparison with an empty tank. This is the best material that was published in the literature a few years before our paper. This is the best simulations we had, and this is the best material that we had in the experimental data, right? Very good match of experimental data, very, very happy with these properties. Okay, this is really interesting, but can we find a deterministic prediction for material performance? Because I haven't answered this question. And the answer is yes, we can do that, but in order to do that, okay, we need to rely on more advanced computational work, okay? And not only Monte Carlo simulations, but now we need to go to these neural networks. So basically just having different descriptors of the materials, we are going to be able to compare Monte Carlo simulations with these machine learning capacities. And what we want is to have all these materials in this diagonal. This is the training, just taking 3 weeks, 1 month. This is how much time we needed to run the simulations. Okay, it's 3 weeks for the oxygen data. They have a fairly good agreement in the training, and this is the prediction when we are running this for 1,000 additional structures. Okay, and you can see we have really, really nice agreement between the machine learning data and the Monte Carlo simulations. And because we know that the Monte Carlo simulations predict experiments, machine learning are also going to be really, really good. What does it mean? 1 minute for 1,000 structures. This means that any new material can be simulated quickly just by knowing the properties like surface area, pore volume, pore size, and so on. Okay, all these ideas are basically we are joining efforts with the Cambridge Crystallographic Database Center, and basically trying to integrate all these machine learning capabilities into their software. So if tomorrow you have a new structure that you have created with your own computer, okay, just you can integrate this into the software from the Cambridge Crystallographic Database Center, and then we will be able to give you some predictions about how materials behave. Okay, next question is, can we really integrate these materials into experimental setups? Okay, all this is about the simulations, and what I really want to work now is more moving into the final applications, and in particular, I want to talk about drug delivery applications. Why drug delivery? Well, drug delivery is really important because in some cases we need to have vehicles to be able to transport drugs. Okay, inside the body, if we have a free drug, sometimes, this is an example that you can see here in the slide, in 10 seconds, we can have the injection. Okay, this is in the leg of a patient. You cannot see the patient yet, but you will see in a second. In just 10 seconds, we can see a very fast distribution in the leg of the patient. Okay, if we wait for 15 seconds, we can see that this free drug, start populating the lungs of the patient. Okay, really fast. In 30 seconds, we can see that most of the blood vessels are already distributing this drug, okay, at around different organs. In 3 minutes, we can see that everything is completely well biodistributed inside the body. And the problem becomes when we wait for 30 minutes. In 30 minutes, we can see the excretion of these free drugs. Okay, this is just one example, but basically what we want to do is to avoid this very fast release of these drugs and the excretion of these drugs. Okay, because otherwise, if we want to have high concentrations in the organs, we need to have very high concentration in the blood, and this means we are going to face a lot of side effects. The solution goes through these metal organic frameworks, and in this case, we are trying to work with these nanometer particles. Okay, we are going to have nanoparticles that are about 100 nanometers with different morphologies, and basically what we want to exploit is all this porosity that we were talking about before, right? You remember all these thousands of square meters per gram. This is basically what we want to have. We want to have the drug molecules getting adsorbed inside the porosity. But also, we want to modify the external surface of these materials in a way that we can improve the performance of the structures. We can improve, we can include some new functionalities, for example, including some pegylation on the external surface, some antibodies, different peptides and different groups that will allow us to improve the performance of all these different structures. We have thousands of combinations, okay, 100,000 different material organic frameworks. We can make a MOF from almost any metal from the periodic table, and we can use all the organic chemistry to be able to modify all these different materials, to play with these different structures. At the end of the day, for drug delivery applications, we want to focus on biocompatible metals, okay? All these spheres needs to be with biocompatible metals, which is going to be iron, sometimes zinc, zirconium, few ones. We will be able to have different morphologies for the nanoparticles and the porosities, and then we can have the storage and the transport of different drug molecules, including RNA molecules, okay, that may fit very nicely inside one-dimensional channels, for example. This is important not only for drug delivery applications, but also for vaccines, for example, right? We have seen these COVID vaccines from Pfizer and Moderna. Why not using MOFs for this application? So why MOFs? Basically, what we want to do is to avoid burst effect. Okay, this is what I was showing in this previous slide. We don't want to have very high drug concentrations and very quick release from the body. What we want to have is something that is going to be more sustainable in time, okay, and something that will not reach very high concentrations, so we can really avoid side effects, toxicity of the drugs. We also want to have selective biodistribution. Okay, we want to be able to target specific organs in the body if we want to do that, and we also want to protect macromolecules from disintegration, from degradation when exposed to, for example, enzymes. Okay, this is something that we have been working on different papers. There's some references here that you can find if you really want to extend the work we have been doing. But going back just for a second into these molecular simulations, right? We wanted to say, well, we have been working quite a lot with gases, but can we also predict drug uptake? Okay, and this is a first piece of work that we published a few years ago. We were working with different materials that we consider biocompatible, okay, made of iron, like these ones, these three ones, also magnesium, potassium, and zinc, okay. And these are materials that you can see the pore size distribution. These materials with different pore sizes, okay. Big cavities, in this case, in the biomorph 100, that would be very important for macromolecules, but also much narrower materials. We can get, similarly to the hydrogen and the oxygen examples, we can also get absorption isotherms for these materials. And in this case, we were focusing on this ibuprofen. Why ibuprofen? Do we need to have a vehicle for this molecule? Well, absolutely not. But there was a lot of experimental data available when we started this study. And that was a good drug molecule that is very easy to simulate and is something, as I said, to compare face-to-face with experimental data to validate all results. So basically, this comparison is represented here in this on the left of the slide. This is the loading of ibuprofen per gram of metal organic framework of material, and this is the pore volume. So in the simulations, you can see we have a very nice correlation, which makes sense. Okay, the pore volume with the loading, and then we were comparing with experimental data that, as I said, was available in the literature. We have for mil 53, we have for mil 100, and for mil 101. We're really good match here, an over prediction here, but the experimental data is the one that looks too low. Okay, this is interesting because, again, we can have a detailed picture of the absorption phenomena at the molecular level, and this is how the material looks like there. This is the crystalline materials with different mesoporous cavities. So something we were doing was trying to block the access of the drug molecules to the smaller cavities that we have here. Okay, we block this porosity, and we can run the molecular simulations again. And in this case, you can see the new simulation was much closer to experimental data. And this is basically suggesting that in the experimental work that was published a few years before us, they were not exploiting all the capabilities of the material. They were just exploiting the bigger cavities that are here. Okay, this is important because after we published our paper, there was a confirmation, experimental confirmation, that you can really reach this loading capacity. Okay, when you have this supercritical CO2 loading, and they were also citing this paper saying, yes, we were able to reach the values that were predicted by the original simulations. So this is for ibuprofen, but we are more interested in real drug molecules, okay, and in particular cancer drug molecules. So just for chemotherapy, we have this very simple one, DCA, which is not really a cancer molecule. It's not a cancer drug, but this alpha CHC and this other more complex one, highly hydrophobic, these are real drugs, including a standard of care drug that you can really find in the literature. Okay, a lot of different papers. We pay attention to the sizes of these drug molecules. Okay, this is the largest size, this is the narrower, the smallest size that we can have in this material. So we can have different values for all these different structures, for these different molecules. And basically, when we selected biocompatible metal organic frameworks with zinc, iron, zirconium metal clusters, we were running these Monte Carlo simulations, and we were getting these different trends that we have here. Okay, this is important because now we can see how molecular simulations can also help us to identify optimal materials, and we can really go for these structures that we have here. Okay, so we can really focus on these materials that we are having here. Depending on the drug molecules, we will say, well, we should pay attention to these mops or to any other one that we have here. Okay, so a lot of flexibility with what we can really do with the computers. We have been extending this, and we are interested not only on the simulation, as I said, but we also want to have experimental data where we can control the performance of all these different structures. Okay, so for this, we focus on two top materials that we identified in our simulations. One is the UIO66. It's a material made of zirconium and BDC. And then we have these PCN 128 and 222 interesting materials with mesoporous cavities. Okay, that's basically the unidimensional channels that we are going to have in the porosity for these materials, the PCN 128 and the 222. Basically, we can load small drug molecules, okay, cancer molecules like SNN38, Permitrex, but also, well, cisplatin, but we can also have a lot of interest in different macromolecules, including this KRAS, which is going to be very important for example, pancreatic cancer. And once we have been able to synthesize these materials, we found that that's not only the only limitation. We also need to reformulate these metal organic frameworks, okay, and this goes beyond the predictions that we can have with Monte Carlo simulations, because we want to have a material that needs to be highly not only biocompatible but also having a very good hydrochemical stability. Okay, if you remember, these materials were not amazing in terms of stability, and something very interesting is that it doesn't matter how robust the material looks in the lab. As soon as we have some phosphate salts, which is something typically that we will have in buffers in biological applications, these materials will get degraded very, very quickly. Okay, so we need to protect them. The idea that we had here is we take them off, we load the drug. So this is doxorubicin, a cancer drug. This material is now red because this is something that will dye the material. And what we wanted to do is to use this weakness that we found in these metal organic frameworks with the phosphate attacks into a big beneficial characteristic. Basically, was taking them off and taking a chain made of PEG, okay, is this chain, but now the head is made with a phosphate group. This phosphate group attacks the external surface of the material, and we are going to be able to incorporate all this pegylation with very, very robust chemical connection. Not only that, not only we can protect the material, we can also include some lyophilization. So we can basically dry these materials, we can get some solid samples that we can really redisperse into the solution. And this helped us quite a lot in the translation of these materials to the clinic. This is basically the materials we were working with. One example with the PCN222, this is the metal cluster, the organic molecule, this is the metal cluster, the one-dimensional channels that we have, and the pegylation. This is how the materials look under the microscope, okay? Beautiful 100 nanometers nanoparticles, some rod shape. And this is how the material looks when it's dried, okay? And you can see everything is very well defined, all these individual particles, and everything is very well connected with these pegylation groups. This is the material in solution, and this is the material when it has been dried under this lyophilization process, okay? This is very important, this is very interesting, because as I said, we can really redisperse all these materials, okay? This is the empty material, this is loaded with doxorubicin, hence the color. And you can see here in the video, we include some sonication, we include the solvent, and we can really have something that is going to be colloidally stable for a few days, okay? We have been doing this for several days, and we don't see any drop, any precipitation of the materials. So very happy with all these different possibilities when including the pegylation into the materials. This is basically the idea. We are going to have our structure. We are going to be able to load this with different drugs, and we just basically pegylate the material. If we pegylate this for too long, we can see that the PEG is able to enter inside the porosity, which is something we want to avoid. But mostly, when we do this in only 2 hours, we can protect just the external surface without damaging the porosity of the materials. This is how the materials look under the microscope, and basically just telling us that in 2 hours, okay, this is the nanoparticle, this is the oxygen and hafnium, which is equivalent to zirconium, and we can see that the phosphorus from the phosphate groups and the PEG is concentrated mostly on the external surface of the material. And as soon as we increase the time, we can see that the PEG has been attacking the center of the material, and it's not only on the external surface. Okay, so basically this means we need to stop here at 2 hours. Can we use this for any MOF? The answer is yes. We have been trying this with different materials, not only with zirconium, but also with different metal clusters. And these are the dry materials, beautiful. And then we can always compare this with experimental X-ray diffraction, but also with DLS to see what are the effects on the particle size. And we can see that there are no changes, which means the materials are colloidally stable. Very happy with this. that was published in JACS this summer. And we have been also doing some testing about the viability of these materials. We can see that non-functionalized materials, they are biocompatible, okay, but when they are pegylated, they are extremely biocompatible. No problems in 72 hours with the cells, and it doesn't matter if it's one of these PCN materials. We have similar results with the different structures. And also, when we load this with the doxorubicin, we can also see that, especially, you can see the difference here with the pegylated material. Okay, the toxicity is lower for doxorubicin than for the non-pegylated material, and even more for the free drug. And the reason is that what we are having here is a very slow release of the doxorubicin. So the doxorubicin is inside the mob, and we just have a slow release, which is basically what we want to have. Okay, we want to improve the performance of the materials by bringing a slower release of these drugs. So just to finalize, this is the final slide that I'm using. Okay, I need to say, well, thank you for the people who is really working in the lab, creating all these different materials, running all the in vitro studies and computational studies to understand how these different materials behave. So, yeah, thank you for everybody in my lab, obviously. Also, a lot of different collaborations in my department around Cambridge, also different universities, and the funding bodies. Okay, most of the work in terms of drug delivery has been funded by the ERC, European funding, also from the Royal Society, and the EPSRC, which is the Engineering, Physical Science and Research Council. So thank you very much as well for you all. Thank you for the invitation and the opportunity to give this presentation, and I will be more than happy to answer any questions. Thank you.
Thank you, David. Thank you very much. I have your camera. I mean, it looks to me it's all black. I'm wondering if it's on. So I can't.
My camera is on.
I can't see you.
Okay.
Let me see. If you switch off and switch on again.
Let me see. Okay, it's on now.
No, it's not coming alive to me. Let me see. Oh yeah, now it is. Yeah. Now
Okay. So if I share my screen, you cannot see me.
Yeah, maybe. Now I can see you.
Okay.
Crystal clear. Yeah. Thank you. Thank you very much. Very good presentation. Very interesting. I was so impressed by the variety of applications you have shown to us. We do have a few questions here. So I'll start with one of our PhD students, Rodrigo. He's asking, let me find here. Yeah, first he wants to know how did you come up, how did you select the force field for all those molecules, and if you use the same force field for all the simulations? So that's the first question.
So that's a very good question, because there are no specific force fields for metal organic frameworks and absorption applications. Or, I mean, few people have been trying to develop a few ones, but there are no generic ones. So we always need to have a balance about performance and flexibility. I think the approach we follow is the more established one, which suggests that we can use generic force fields. And in our case, for the metal organic frameworks, we use Druiding force field. And this is something that works well. Okay, it has obviously some clear limitations, but it's not too bad. If you are missing an atom from the structure in Druiding, then we use universal force field, which is also a possibility. But universal force field tends to over-predict interaction with the molecules we want to absorb. And then for the if we are thinking about gas molecules, well, there are Trappe force fields, for example, which are extremely good. Okay, all these force fields are really accurate. As soon as we go to more drug molecules, for example, then we need to build the models for these structures, and we try to use a combination of something like OPLS, for example, to be able to get this. I don't remember on top of my head, but there's some software that can also help you to define the models for drug molecules if you are interested. This is something that we have used in the past, which is basically you just draw the structure, you saw this, and it's able to give you some ideas about the different angles you need to take into account, and all the different sections in these molecules. And this simplify quite a lot the molecular simulation process, at least preparing the force fields and models. Okay, we use a software for Monte Carlo simulations called RASPA. This is something that was developed between the University Pablo de Olavide in Spain and Northwestern University, and that's basically the idea. Well, not only these ones, this also with David Aveldam, for example. He's in Netherlands, so it's
Yeah, good. Another question is how did you integrate the crystallographic database with the molecular structures for Monte Carlo input files?
Can you repeat the question? So how can we really compare?
How do you integrate the database data with the molecular structures for the input file in the Monte Carlo simulation?
Okay, so basically you can download now. Okay, it doesn't matter if you have a license on the CCDC or not. We had a long discussions with them about having access to a open source database of structures. So they agreed to publish three-dimensional metal organic frameworks, and this is a selection of about 15,000 different materials. This is something that you can download directly from the CCDC. Okay, it's very easy if you Google for that. It is something we also publish in Matter this year, I believe, and you can get a file with all the CIF, okay, all the crystallographic information files. You can put all this folder into your molecular simulations software package, and then you can really have some scripts to be able to read all these CIF structures and be able to run the simulations. So that's very easy to implement.
Another question goes like this. What are the limitations of the prediction models for screening of MOV's performance?
It depends on what application, obviously. So if we are thinking about gas absorption, okay, Monte Carlo simulations can predict a lot of information about uptake, working capacity, interaction. You can even use molecular dynamics to know about kinetics. So there is a lot of information you can really refine. You can start moving into the process simulation side of things, where you can really start integrating this with all the kinetics information, so you can really expand that. And this works. Okay. The problem is obviously when you work on drug delivery applications, this is so complex problem that things get much more complicated. So the molecular simulation so far will allow us to identify some optimal materials, but we need to go to the lab and we really need to check that the materials are biocompatible, that they perform well in the drug delivery, they are really able to cross cell membranes, and we have a proper endocytosis pathways. Okay, we have been working on this for 10 years now, and obviously we have a lot of challenges that we cannot really predict with the supercomputers. So it is always a very important tool in the process, but it's not the only way of getting information.
Regarding the usage of MOVs as drug delivery systems, is it possible that the MOFs build up in certain parts of the human body?
If they can accumulate?
Yeah.
Yeah. So yes and no. There are some papers that suggest that some materials, for example, I remember from Professor Patricia Orcajada, she was working on MIL-100 iron, and they found that it tends to accumulate in the lungs, okay, but then the material dissolves and the body gets rid of the MOF. So there is a short-term accumulation, and that could be interesting because if you are looking for lung cancer applications, for example, or something related with, I don't know, asthma, perhaps, you can use this to as a benefit, something that you can really exploit. If obviously the material really accumulates at the end of the day in the lungs. That will provoke different problems, right? We focus on one of the cancers we focus is mesothelioma, which is provoked by the exposure to asbestos fibers, and all these fibers will basically start producing this cancer 20 years after the exposition. Obviously, if we have something similar with the mops and they really accumulate in the lungs, they can really provoke these kind of problems. But as I said, the materials are not that stable, okay, and this is very good. This is in contrast with carbon nanotubes, gold nanoparticles, and so on. So there will be a little bit of accumulation at some point, especially if we include targeting agents, okay? But as soon as we wait, in less than a month, the materials dissolve completely.
Okay. I have one question myself. Maybe it's a silly one, so if you accept my apologize beforehand. It's just maybe out of curiosity. When you were explaining, when you're going through the pegylation, if I may say, time, in order to avoid that you have your substance in the porous region, so then you come up with some time. So the thing is there any particular simulation that you can do beforehand to try to come up with what is the optimum time, or is just something like try and error?
I think that's a very good question. We included some molecular dynamics simulations in this paper, but at the end of the day, I try to be very pragmatic with the computational work we do, and to me, computational questions are important when they help us to resolve problems. Okay, if it is faster to solve the problem running the experiment, let's run the experiment. Okay, to me, the point of using the molecular simulation or any other simulation that we are using is because we are going to be able to solve problems, anticipate problems, and go through that. Maybe somebody will be able to develop some proper processes to simulate this pegylation process more accurately. Okay, in our case, it was much easier just to do trial and error. We test different times, and then we see which one was offering the best results. Okay, simulation for us is a tool, but yeah, we always use this in combination of experiments. In this case, experiments. We felt more comfortable with experiments. But it's a good question.
Yeah. Okay. There's one thing that was following your presentation, as far as I could understand, when you were showing us the comparison in between the experimental data and the computational data. It sounds to me as if it was like a blind test. You didn't know beforehand what's going on, so you got very surprised when you got such good agreement. Am I correct? Yeah. That's the impression that I have.
Yeah, we don't get very surprised. Okay, we were expecting good agreement. Some materials have excellent agreement. It is true that there are other simulations where the agreement is not that good. Okay, we also understand that when we are simulating materials with very open porosities, okay, this is very related with the question with the force fields. So when the force fields are perfect, the simulations work really nicely. And in a force field, you have two, we use Lennard-Jones parameters, and we will have two main terms. One is going to define what is the size of the atoms, and the other one is going to define what is the interaction of the atoms. The size of the atoms is generally really well defined. So capacity of materials, the simulations work really accurately. Okay, if you don't have defects in your crystalline structure in the experiments, then the simulations will work really well. The main problems are related with the interaction of the molecules. If the structure is very open, all this is mainly dictated by the gas-gas interactions, for example, okay, or drug-drug molecule interaction. But if we have a very narrow porosity, then the interaction between the MOF and the guest molecule is going to be more important. And if we don't have specific force fields for the materials, it means that we will fail probably on the interaction. This is where I say we tend to overestimate the interaction of the gas molecules with the MOFs. But the gas-gas molecule interactions in particular, they are very well predicted by these TRAPE force fields. And this is really good. This is something we can use. And this is when the simulations work extremely well.
I see. Yes, let me see here. I don't think we don't have more questions, at least here. Let me check on the YouTube, just one sec. Yeah, maybe if any other question come up late, maybe I can email you them And
Absolutely.
yeah, and once again, I'd like to thank you very much for accepting our invitation. It was a very good presentation. Also, the opportunity to chat with you and discuss some of the research you've been doing. Congratulations for your work. It's very impressive, honestly. And,
thank you very much.
Yeah, and I think lots of people here maybe can get in touch with you in the future for some sort of collaboration. We have a strong team in our department which work with the molecular dynamics as well and computational. So probably someone will try to get hold of you at some point, I suppose. And again, I'd like to thank you very much for accepting our invitation. It was very good contribution. And I don't know, I am myself alumni from Cambridge. I'm from St. Edmunds, and maybe I still have some collaboration with these people from the fluid groups in the energy, not in the ChemEng, but used to be the other side of the road. Now I know that you guys moved to West Cambridge, right? So it's
we moved to West Cambridge, so we have a nice new building. As you mentioned, also working with Ian Wilson, I believe. So
maybe I have plans to be in Cambridge next year to visit Stuart. So maybe I just pop in and we can have a cup of tea or something.
Perfect. That would be great. I mean, this pandemic is obviously offering a lot of flexibility with these presentations, and this is great. But if you are around Cambridge, I mean, delighted to obviously talk to you.
Yeah. Okay. Lovely.
Thank you very much again for the opportunity of talking. And I do apologize also for being late because I was really very confused with the time used. I'm very sorry. But thank you for the opportunity.
That's one of the things we've been receiving guests from around the globe, and sometimes we get confused with these time zones. So that's absolutely fine, absolutely okay. Yeah, what a great pleasure to have you here. Thank you very much.
Thank you very much. Thank you, Xavier.
Okay, you take care.
Take care.
Yeah, bye.